So watch lead and those watching at home, just so you know, on the quad split, I've modified the 4 displays. Stream 1 will still be the ROV. On stream 3, the upper left quadrant will be the ROV. Upper right will be the ROV's graphical user interface, so you can see the depth as well as the direction. The bottom left will continue to be the high pack with the direction the vehicles are going, but the bottom right is our EK60 readout.
Thanks, Roland. Is it possible on the bottom left to put a CTD trace instead of the seafloor bathymetry? I just did turn my mic on, didn't I? I'm on the upper one. Thank you. Yes, please. Okay. 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 Let's leave it as it is then. Okay, thank you. All right, so we're starting the transect right now? All right, thank you. So, all right, so for anyone on the line now, you might have noticed that we are doing different. Okay, is that better? Okay, so for anyone on the line now, you might notice that we're doing some pretty different operations from what we normally do as we start to recover the ROV. So today we're going to be doing some midwater transects, and instead of just transiting right through this large volume of water that we typically transit through, we're going to look for some of the very interesting and rare critters that live out here. So my name is Amanda Netburn. I work for the Cooperative Institute of Ocean Exploration, Research, and Technology, and work. So we're seeing something here. This looks to me like it could be collapsed larvation house. Let's see if we can get a closer look. Yeah, actually this one to the kind of lower middle part of the screen might be more interesting. So this. What's on here? I do not actually know what that is. But we will send that around to some of our other Midwater experts if we have anyone else on the line who wants to chime in. Thank you for getting that Zoom. Yep, so the other folks we had on the chat didn't know what that was either, so we already have had a good example of kind of how little is known out here. So the plan today is we're going to be doing 10-minute time transects at 7 different depths in the water column. These will be at 1,800 meters, 1,500, 1,200, 900, 700 and 500, and then we're keeping an eye on the EK60 water column backscatter data, looking for deep scattering layers, and we have one more target depth that we can pick based on what we're seeing.
So Amanda, have you had given an opportunity for the folks on the line to introduce themselves?
I have not yet, so anyone who's on the line, if you can go ahead and introduce yourselves. I believe Astrid is still on.
Yeah, hi, this is Astrid. I'm at the UHECC, and I'm staying on for these midwater dives.
Fantastic. What is your background and expertise, Astrid?
I'm actually more of a seamount ecologist, so I don't work too much in the midwater. But I do specialize on deep sea fish, so hopefully we'll see some fish I can recognize.
Awesome. Thank you for joining us. And then I know Dougal was in the chat. Is he on the phone by any chance, Dougal? Sounds like he has not been able to call in. I think he's in the middle of travel and trying to squeeze this in. Probably limited capabilities. Is there anybody else on the line right now?
Yeah, Amanda, this is Chris Kelly at UHECC2. But what I'm going to do is I'm going to try to shoot home right now and then fire it up from home and watch the rest of the Midwater transects from there.
Okay, sounds great. Thanks, Chris. So right now, all right, so we do have one more person participating in the chat who has not been able to call in, and that is Dougal Lindsay, who's a professor at JAMSTEC in Japan. And he is, I don't know, somewhere in the middle of travels all over the world. But he is an expert in these gelatinous animals, which most of what we'll see today are likely to be gelatinous animals. So unfortunately, Dougal is having difficulty getting enough bandwidth to stream the video, but fortunately we record all of this, so we'll be able to make some good zooms on all the organisms that we see, and we can send him that video as we complete the dive and do our annotations afterwards. So you'll see right now most of what we're seeing are just these kind of white specks floating by us. This is what's called marine snow. So basically everything that's up in the surface of the water column where all the primary productivity is, where a lot of the life is, slowly decays and forms these little particles that slowly rain down to the seafloor, which is why we call that marine snow. If we can zoom in on this guy. So we are seeing here is a larvation towards the kind of lower left. You can see a long strand of something. It's a small tadpole-looking animal. It's called a larvation. Sometimes it's beating its tail, so you can actually see some movement in there. The rest of that structure is what's called a house, and it's a filtering structure that allows the larvation to filter out small particles, which is what it feeds on. these houses clog really often. You can see how fine it is and how many particles are already stuck to it. So every so often, and this can apparently be a couple times a day, they will shed their house and build a whole new one. And now you can see it wiggling out of its house. So we got to see exactly what I was just describing there. Ditched its house. And now you can see the little tadpole larvation swimming around. And what I've heard is it can potentially take maybe minutes to build a new house.
That is awesome. I've never actually seen that before.
Cool.
Did you say several times a day?
Several times a day.
Yeah.
So one of the major sources of this marine snow that we're seeing are these abandoned larvation houses. And you'll often see as they sink, and we might see more of these as we get closer up to the surface. As they sink, they'll look like a big teardrop shape as they sink down to the seafloor.
Oh, there he is.
Really great. This is fantastic. So as you can imagine, you saw how delicate that house is. It's really hard to study these animals and how that house works, because as soon as you try to collect it, the house falls apart. You can collect the larvation, but not its whole feeding apparatus. So there are some folks at MBARI, the Monterey Bay Aquarium Research Institute, who have been using these like laser planar scanners to look at the way that particles move through those houses. So without having to disturb the animal or disturb the house, they can actually visualize how they're feeding. It's some pretty amazing and cutting edge work that's happening there now.
I see those, and I always wonder how all the old textbook artists came up with these just amazing drawings of these things.
I know.
It's like, wow, how did you get it to sit still?
I know, it's really true. Let's see what we've got here. I have no idea what that is.
That's interesting. It looks like the head of a copepod or something.
Is it possible to get any?
So this looks like a medusa that's oriented sideways, and then some kind of crustacean that's hanging onto it.
Oh, it's not transparent.
Oh, is that a Hyperion? Yeah, that might be a Hyperion amphibod. Those are the eyes. Those are giant eyes, that whole anterior part. I'll bet that's what that is.
They're normally quite transparent, but
sure.
But I bet that is what it is. So these Hyperion amphipods can parasitize salp barrels. So basically go in and live inside them and take over their homes. All right.
Oh, so that was? Okay.
Excellent. Thank you so much. So it may have seemed like we didn't see all that much at this particular depth. But we're quite deep, and the pelagic fauna are really few and far between at these depths. So it's pretty exciting that we've gotten to make these observations. No one's really ever looked for these kinds of animals at these depths before. As we move up in the water column, it's likely we'll start seeing a lot more organisms, as well as more of the marine snow. So we can see in the EK60 backscatter data that there is a pretty thick layer from about 300 to 500 meters, and then a second layer below that around 600-700 meters. So what this backscatter data is we send an acoustic ping out in the water column, if anyone's been listening, about how we do the mapping on the seafloor. This is very similar technology, just at slightly different frequencies. anytime there's something in the water column that has a different density than the water itself, so crustaceans, things with hard parts, fish that have swim bladders and their bones, all of those things will reflect some of that sound back up to the ship. What that gives us is a really nice profile of the water column. There was another larvation house. We got a really nice profile of the water column, and we can see where most of the animals are living. So again, at 1,800 meters now, we're far below that thick layer that we're seeing and towards the upper water column.
So Amanda, as we are transitioning, I have one of my favorite questions for you. Why is it important to explore the water column?
Great question. So for one, is just kind of this fundamental understanding our planet. The water column is one of the least explored parts of our planet, and yet comprises some 95% of the inhabitable volume of our planet. So really everything that's a great percentage of what's happening on our planet is happening in the water column. So it's fundamentally important to understand what kinds of things are living there and what sorts of processes are taking place. But there's some other more kind of relevant practical reasons to understand the water column. So this supports many of our fisheries, tunas and swordfish, other of these large migratory species all use the water column throughout the globe. Also marine mammals and sharks and other large organisms of conservation concern are using the full water column. And then also, again, this marine snow, it might not look that interesting, but it turns out that the carbon that is taken up by the ocean eventually makes its way down to the seafloor through this marine snow. And so it's very important to global carbon cycles to understand how organisms are taking up that carbon energy and where it's eventually going. It has huge effects for the planet's climate and other processes, connection with land and atmosphere.
I was on mute, sorry. So one of the things that we talked about earlier today during the benthic dive is that a lot of what we are doing over the last couple of days has been in the high seas. So we talked about the importance of studying the high seas and why we spend time out here when we're an American vessel. So why don't you talk about a little bit about
Sure.
Why would we be exploring the water column in the high seas and how is that different?
Sure. So again, there's some of the relevance with fisheries of concern. So for things like tunas and other animals that come into our waters. So in the water column, there are currents, there's upwelling and downwelling. So no little piece of water stays exactly where it is. So it's actually moving in and out of different nations' EEZs, their boundaries in the ocean. So it's all really connected. So what's happening outside of US EEZ is still important to what's happening inside of the US EEZ. Another reason is that there are really no programs dedicated to studying the water column on the high sea. So the International Seabed Authority regulates the seafloor and resource use on the seafloor. There aren't really comparable things in the water column, so we don't really have people ever going out here and looking and trying to understand it. So it's a pretty incredible opportunity.
So while you were talking about the EK60s, Astrid brought up that sometimes we see a vertical migration there. So Astrid, would you mind telling us a little bit more about what you were saying in the chat log?
Sure. I was just talking a little bit about deal vertical migration. So I think you mentioned that this is the largest mass migration on our planet and actually happens twice a day. So these are made out of animals. This layer that migrates is made out of animals that will hide at deep, dark depths during the daytime and then migrate up the water column to the surface at night to feed. So we're at about coming up on 4 o'clock, I believe, Samoan Standard Time. Is that right?
Yes, that is correct. We're about 3:45.
Right. So then we're still probably seeing these animals at their daytime depths. Once the sun starts to set, you can watch in the EK60, you can watch this layer kind of rising up towards the surface, and you can see it really clearly in the EK60s. So it's a fun thing to be able to watch or look at in that data set. So we're going to be actually looking at kind of their daytime depths of this layer. So that'll be neat.
Cool. Thanks for that explanation, Astrid. We actually saw that this morning. We had the EK60s on early in the morning, and we could see very clearly that whole layer migrating down as the sun came up, and as you're saying, hiding away in the darkness of the mesopelagic zone.
So in stream 3, I just momentarily put that up full screen on the EK60. That slanted line on the bottom right is the
oh, yeah, that's great.
ROV.
Thank you.
Thanks, Roland.
So one of the things you can see if you look in detail at that EK60 that was just up is that you can actually see that there's distinct sublayers within that big, most obvious scattering layer. So it's not quite as simple as we thought maybe in the past, where we've kind of viewed it as one solid layer that would move. There's actually different sublayers within that one scattering layer, which you can see in that EK60 graph.
Yeah, so another interesting observation we had earlier today, because I've been keeping my eye on it most of the day, when the ROV first went through the deep scattering layer, when we were in that kind of upper, thicker layer, the animals didn't really move away. The layer remained, but as the ROV made its way closer to that deeper layer of the larger piscivorous fish, they started to go down, and then as we got a little closer, they just darted away, and we stopped detecting that for about 45 minutes, and then finally the ROV was enough below that layer that we started to see it pop up again. So that was pretty interesting to see.
Yeah, avoidance can be tricky with the ROV, especially at mid-water depths. So generally, I think the way that mid-water transects are done is on the descent so that you get into undisturbed water. So we have some challenges here doing it on the ascent.
Yeah, I'm very interested to see, and we'll have the EK60 on this whole time, how the avoidance differs on the way up compared to what we detected on the way down. So, and something I've noticed talk to other colleagues is that it can change from location to location and day to day what kinds of behaviors you see of the organisms when an ROV comes nearby. Sometimes you'll see a aggregation of fish follow you around for the whole dive, and the next day do a dive in the same place and not a fish in sight. I've talked to Bruce Robeson at Embari, who's been doing this for decades, and I said, have you figured out, why one day it's this way and the other day they won't come near us? He's like, I have no idea. So we still have a lot to learn about what they're doing.
Yeah, I think we saw some of that too down when we were on the seafloor, where we saw some of the animals getting really confused by the ROV, and some of them kind of darting away and swimming away, and other ones kind of frozen like deer in the headlights, and some coming in to check out the ROV. So it definitely seems that like different species are having different responses to the ROV.
Absolutely. So yeah, as we were saying, the fish, we know tend to avoid the ROV, but we have other ways of collecting a lot of the fish that live at these depths, which is using large trawl nets. For the gelatinous animals, which fortunately do not avoid the ROV, they tend to get really mangled in the nets. So that's really one of the least known parts of what's down here, and something that we're a gap that we're really able to address by going down with the ROV and looking around, is to see what the distributions of these different species of gelatinous animals are. There's, our colleague Dougal, who's in the chat room but not on the phone, has discovered multiple species that have been undescribed before of these gelatinous animals by doing this kind of work.
So Amanda, what's our next target depth for the next transect?
1,500 meters. So we're getting pretty close here. I think yeah. So the last one I see is 1544.
Yeah, see right there.
And is there any way we can get the EK60 back up on one of the quad screen?
It is up on the quad screen. Mike is just having to reset it. A button was accidentally pushed to clear it out, so Mike just reset it up. So what you're seeing is it's actually streaming from the right-hand side, the live data.
Oh yeah, I see that. Sorry.
Okay. And also, if you guys wish, I can make it full screen, but I thought the having the depth of the ROV and the position of the map would be useful. But if you'd like, I can make it full screen.
Astrid, would you prefer it full screen? I know that the map for this stuff is not too important because we're a ways off the seafloor. So any preference, Astrid?
Yeah, I think it might be nice to have it full screen.
Okay, we'll get that up for you.
It's done.
I can hear you. Who are you talking to? Oh. Just watch lead one.
Okay. Did something happen, or? No. Okay. Just got slapped at me.
Sure. Let's go with that.
Okay, we'll talk.
Anyone's coming in now.
Okay, thanks. Keep hitting the wrong button.
That's you. That's you. The pilot's talking to you.
Oh. Yeah. Hi. Okay, thanks. All right, so we've just reached 1,500 meters. The pilots are changing, and then we'll get started on our next transect.
So Dougal Lindsay just wrote a comment in the chat room that there actually is a second group of predators that will migrate shallower during the day from the bathypelagic that goes up to the mesopelagic to meet the DL vertical migrating fishes that are then coming down during the day. So I think there's a lot of dynamics happening with the DL vertical migration that really makes it actually quite complex, and we still have a lot of work to do to really understand what all is going on there.
Very cool. Thanks for sharing that.
Did you make a note that we started this?
Have we started?
I think we just started.
Okay. All right, so we are starting our 1,500 meter transect.
Is that a shrimp there on the right?
Maybe an isopod. I think I see some long limbs. Or a copepod.
Yeah, looks copepod to me.
Yep.
Yeah, big copepod.
Yeah, I was talking about something further right that kind of drifted past out of view already.
Can you still see it, or?
Come on, there's no crustacea.
No, I think that's gone.
Water column inverts doesn't have any crustacea.
Oh. Yeah.
What's an oversight?
Yeah. You could just type it in.
I did.
Oh, nice.
So maybe a
Chetignath, yeah. So these are also known as arrow worms. So in every marine biology class I've ever taken, these are referred to as the voracious predators of the plankton. They have really large jaws that open up to their sides, and they can eat things that are, relatively speaking, very large. But if you actually collected one of these, took them up, the biggest ones I've seen are maybe a couple centimeters long, so they're not quite so scary in person.
Under a microscope, those grasping spines are pretty good.
They're impressive.
Yeah. There's something.
All right.
Something with an egg sac or an emerdian?
Shrimp, yeah.
Yeah.
Shrimp.
So maybe a mysid, maybe? Whoa.
Could be mysid.
Here's something.
This
Maybe
looks
another
like
larvation.
another larvation house to me.
Yeah.
And I think someone's home. Only seeing one lobe of the house. About this here in the center. Alright.
So Dougal suggested for that copepod that we saw earlier that it might be a megacalanus or a bathycalanus.
Okay.
And then he also commented that those can be cannibals.
The Chetignath or the
Copepod.
copepods? Okay. That's harsh.
Yeah. There's something right here. Maybe another Chetignath, maybe?
That would be my best guess without getting a better look on it. Yeah.
They're voracious predators and acrobats. I
Does
didn't look.
not
Oh,
look
this is
particularly
something now.
biological.
There's something
Yeah,
right
something
in the
right
center.
in the middle. This is the same thing that we saw earlier with these spikes on it.
We saw some interesting spiky thing in 2015 that Steve Haddock identified, and I'll have to look on my computer, but it was a protist of some kind. I was so surprised.
Yeah.
Like a radiolarian or something crazy like that.
Nice. Little jelly.
Yep.
So this looks to me like it's one of the hydromedusa. So they have a ring around the opening that's muscular.
Valerium or the vellum.
Vellum, that's what it's called. Oh yeah, this is definitely hydromedusa. And I'll need to pass this along to one of my gelatinous expert friends to get a full identification on it. It would be great if we can get a good shot on it.
Kind of looks like
really
a
beautiful
coronate, though. Looks like it had a furrow on a kind of pointed top.
Oh, something else. All right, thank you. Yeah, that's good. Thank you.
Or a larvation, maybe. Whoa, let's slide.
Potentially.
Former house.
I think so.
Yeah.
It almost looks like it could have been a low-bait Siphonophore, but I think it was a house. Okay, so that's correct. Thank you.
At the end there, it looked like it could have been a cestid Siphonophore. So either low bait ripped or the cestids are the ribbon ones.
Yeah, yeah.
That's what I thought I was seeing. Periphylo.
Yeah. So those are a type of hydromedusa, aren't they?
No, those are Scyphomedusa.
Oh.
You can see the little furrow between the top and the bottom of the bell. But I couldn't tell if I actually saw indented margins.
So Dougal said that would have been pretty deep to see a cested ctenophore. So if it was a ctenophore, it was more likely a ripped lobate.
Mm-hmm. What did you want?
Something falling down. There's a fish.
Yep.
Are you able to? Yeah. Looks cyclothony to me. Astrid, did you get a glimpse of that by any chance?
I really just got a brief glimpse, but
Okay.
not a good focused-in shot.
Okay.
Cyclothony might be a good first guess, but I didn't get a good look at it.
Yeah, that would be my best guess based on what we were seeing. So cyclothony the common name is Bristle Mouth. They're rarely seen. Very few people know they exist, but they are actually the most abundant vertebrate on the planet. So of everything with a backbone, all the fish, all the mammals, all the people, everything else, if you counted up all the individuals of any one taxon, there are more cyclothony than anything else. They're quite little fish. They don't get more than a couple inches or so big. A colleague of mine refers to them as a backbone with some skin on them, which is pretty accurate. There's not a lot to them. But the The deeper cyclothoni do tend to be more more muscular, larger, and more pigmented. The ones that live in the really shallow water are, or relatively shallow, I should say, water, are what we call pedomorphic, which means they retain the characteristics of the larval form. And the use to them of doing that is they're small and they're pretty transparent, which is what a larval fish looks like. So it helps them basically camouflage and hide from predators, which are far more abundant in the shallower water and are using light to find their prey. There's a lot of stuff darting around. Mm-hmm. Yeah. We've been getting some questions in the chat room about trying to size things out here. And the tricky thing is with, if we shine the lasers out, there's really nothing for them to reflect off of. And most of the organisms that we see out here are smaller than the distance between the lasers, which is about 10 centimeters. So it's very challenging with standard tools to actually size things that are in the water column. We're imaging a three-dimensional volume as opposed to kind of cruising along the seafloor, and we don't have that background of the seafloor to reflect the laser lights off of.
So if that fish that we did see earlier was indeed a cyclothony, there are some cool things that we can talk about cyclothony as a group, or the bristle mouths as a group. Something like they have very small eyes, actually, which is interesting because you'd think that things need big eyes to see, but the idea is that they actually use their lateral line and other senses more than their eye. They also have they also can counter-illuminate. They have photophores. They can produce light, and that is thought to be a strategy to hide their shadows from predators, since these bristle mouths are really a big prey item for a lot of other fishes in the mesopelagic. So they are really a pretty neat group.
Great. Thank you so much for that information, Astrid. think so. I don't see
No.
bracts.
Maybe, is it just
Could have been larvation
squid?
house or squid ink, you think?
Yeah.
Yeah.
Pseudomorph.
Ctenophore. So that was a ctenophore. You can see it's kind of a little bit mangled, but you can see those lines along it that they look like they're producing light. They look like a little rainbow, but it's actually a row of cilia. They're called teens, and every ctenophore has 8 of those rows of cilia on them. They can have very different body shapes. Some are really round little kind of ping pong ball shaped. Some are flat and long, but every ctenophore has those 8 rows of teens or combs. The common name for ctenophores is comb jellies because of that feature.
So an interesting note about ctenophores, a lot of people get them confused and classify them in the same group as jellyfish, but they're actually their own phylum. So that's always an interesting note to make for anybody who's tuning in from home. They're actually a totally different phylum. They're not in the phylum Cnidaria at all. They're in the phylum Ctenophora.
Thank you, Astrid. Another point on ctenophores that Dougal typed into the chat room here is that we're still finding new families and even new orders of ctenophores. So again, this really speaks to why it's so important to explore out here, is that we're still finding so much that we didn't know was here before. All right, so we are starting our 1,200 meter transect.
Can't tell if that's 2 stuff on the. Oh, it's gone.
One left. Possibly. That looks just like some particulate matter, so that could be a collapsed larvation house or some other bit of sinking matter. Marine snow, when you get close to it, can take a lot of different forms. There's an instrument called a laser optical plankton counter, which basically just gives you like a black and white image of any little particle that comes through it. And we find it's remarkably difficult to distinguish between zooplankton and particles sometimes because of all the different shapes that this marine snow can take. Also looks like it's just sinking matter. Right to the right of the... Yeah, this is more marine snow.
This is moving now.
Yeah, great. It's tiny.
Moves kind of like a crustacean.
Yeah, I'm definitely thinking crustacean.
Little
maybe
bursts.
some kind of amphipod. Is that as close as we can get to it? I know, it's really hard to tell.
We may have passed it already and we don't even know it.
Yeah.
I don't know, I think it's copepod the way it's jerking around there.
Yeah. So, so you've probably noticed already that the marine snow is already starting to get a bit thicker, and we're starting to see more of those kind of elongate forms of it. So what happens is as the marine snow sinks down to the seafloor, bacteria glob onto it and start decaying it. So as we get deeper and deeper, the marine snow particles get smaller and smaller and fewer and further between as the bacteria convert it back into dissolved nutrients in the water column. So we'll continue to notice this as we make our way up in the water column, that there will be more marine snow, larger formations of marine snow.
Something, yeah, on the left of center. Something over there. No, it's moved off screen to the left. Zoomed past it. It's kind of a string. It was a glittering string. Okay, don't see it.
Yeah. Oh, wow.
Oh, yeah.
So,
yeah.
is that one of those, yeah, I think so.
Pelagiotheria.
Yeah. Yeah, I can get that in. It's another little crustacean coming through. Disappear.
It's kind of amazing. The pilot is kind of, oh yeah.
Oh, yeah.
On the starboard side.
So this is a pelagic holothurian. So if you're familiar with sea cucumbers, they are an echinoderm related to sea stars and sea urchins. They are almost entirely benthic, but exists this type of pelagic one. You can see how it has this sort of umbrella-like shape to it that helps it float in the ocean and kind of maintain its position up in the water column.
You can see the tube
really
feet.
Cool looking.
Yeah, the tube feet just restricted to around the mouth. And ironically, this is the first holothurian we've seen all day. We saw none on the substrate. How bizarre is that? Great.
That's
great
fantastic.
Find.
Yeah.
Yep. Good eye up there, whoever saw that, Chris
yeah.
or somebody.
So anyone watching this at
That
home,
was beautiful.
isn't that cool?
Yeah. Agree, Astrid.
Anyone watching this at home, you know, you just get a couple of the streams, but the pilots here have, I don't know, a lot of cameras that they use to navigate around and keep an eye on all their different systems. So they often are able to find things that we wouldn't just see in the main cameras. Pelagothoria. So for anyone who hasn't been following along on the chat, we've had a request. So this is to please keep our eyes out for a dish-like medusa with 32 long tentacles. It's called Solmysis. Our colleague Dougal Lindsay believes that they are flowing northwards from the Antarctic, but they don't have any records yet for this area, only off Japan. So this would be really helpful to help connect the dots and understand some of that connectivity between the populations of these species. like a Kawhiti. Off. Something in the middle right, maybe.
Is that a Medusa?
Maybe.
Oh, you know, this might be.
Almost looks like a salp.
Does almost
Like a
look
dead salp,
Yeah,
you know?
I
Yeah.
just see a scalp out loud, but I don't know.
Kind of
It's
looks like
really-
it, but I don't know that it's alive.
Yeah, I mean, whatever it is, it seems to have a lot of particles on it. This makes me feel like it is not particularly alive. Strange. There's a Kawhiti in the background. All right, I think this is good. We can keep searching. Thank you.
Is that something that's just going across center now?
I think this isn't going to be anything. Yeah. Marine schmutz.
Yep.
Thank you. Something just zoomed across there. Thank you. So we have 2 minutes left of this transect. This depth.
There's something red right at the top of the screen going across to the right.
It's moving towards
Almost
upper
center.
left.
Yeah.
Yeah.
Small, huh?
Yeah.
Maybe it's a larva. Wow. It's really small.
Yeah. Amphipod to me, or copepod.
I think copepod.
Yeah.
Yeah, I think that's another copepod.
Yeah. They are, the deep copepods are really chubby, for lack of a better word. They have a lot more girth to them. Yeah, it's gonna be hard. These guys are small. Yeah. Thank you. Oh, that lighting looks good. All right, sounds good. So we are ending our 1,200 meter transect. We are headed up to 900 meters next. 900 meters. Astrid, are you still on the line?
Yep, I'm still here.
Do you have the EK60 screen up?
Yep, I can see it.
Okay, so maybe you can do a little brainstorming with me. So what we have for our next few transects are 900 meters, 700, and then 500. And then we have one more that we can kind of play around with to target the deep scattering layer. So I'm wondering, it's a pretty thick layer, but wondering if you have any
that
thoughts.
layer is right at like what do you think that layer, that green layer is right around like 450 maybe?
Yeah.
So if we were at 500, we might be just at the bottom of it. So if we could move it up a little bit to go through that green layer, that might be interesting.
Okay. So the other thing we could do is do like a 600 and then a 450 or something instead of the 500 and the last one.
Yeah, so the other thing I can see on the EK60 is that like maybe around, there's like two blue layers with like a gap in the middle.
Yeah.
So it'd be interesting to do one transect through the bottommost blue layer and then another transect above that and then a third one through the green.
Okay.
Are you talking about the thicker blue ones, Astrid, that are below the green, or the ones above the green?
Below, the two blues below, but there seems to be like a distinct blue layer down at maybe around, I don't know, maybe 800, and then there's a gap, and then there's another blue layer that starts, I don't know, maybe around.
We can see it, Astrid.
That green layer on top of that. So it'd be kind of neat to see if that bottommost blue layer is actually different things.
Yeah.
That top layer, since it does seem to be clearly separated, which seems interesting.
Yeah.
Yeah, I agree. I was going to suggest the same thing.
Okay.
Two layers.
So you think maybe move the 700 meter one to maybe 750 or 800 meters?
Oh, there it goes.
Well, I just want to show you. So it's 1,000. It's 500 up there.
Hard to tell the depth on this display.
Can you repeat that?
I was saying it's hard to tell the depth from at least my home computer. This is Chris, by the way. I made it home and logged on.
Oh, great. Welcome back, Chris. Nice to have you on. Yeah, the depths are a little tricky. I think our 700 will get us pretty well into that deeper layer.
Yep.
So, yeah, which is right where that deeper layer is.
Yeah. Maybe even 705 or something, just to make sure you're, in and not at that break.
Right.
705, 710.
Okay. I suspect some of these things
While
will
we
move
were talking,
around
I
as we
think
approach
maybe a Cerebomber passed by. It looked like a long, elongate eel-like fish hanging in the water column.
I think that was Siphonophore that we saw just a couple of minutes ago, Astrid. Is that what you mean? Oh, there's another Pelagotheria just went by.
There goes another hollow. Yeah, I mean, it was a ways back. It was like just kind of a line that passed through real fast, and I thought maybe it might be some sort of an eel-like fish.
That's definitely possible. I was looking pretty closely at the EK60, so I might have missed that. Sure, let's just solidify our plan, and then we can get to that. All right, so we're thinking 710, and then 450, and then we have one more we could play with. So we could do like 600 and get kind of the bottom of the upper layer, or we could try and target a shallower one, which kind of gets into the range they don't want to be in. So what do you think about doing 710, 600, and then 450 as our last three after the 900 meters? Does that make
That was
sense to you?
710, 450, and what was the other one?
600. So that gets us into sort of the deeper part of the upper layer, and then the 450 will get us into that really core yellow area of that upper layer.
Yeah, that sounds good.
Okay, awesome. Thank you. So Mike, if you want to explain to us a little bit what's going on in the EK60.
So for those at home looking at our EK60 data here, other than the layers of biomass that we see at the top of the water column and the stepped ROV track that we can now see, you should be able to see some just straight up and down vertical lines indicating some sort of electronic noise in the water column. And this morning, in communicating with our ROV team, that is actually the noise or the signal that's showing up in our EK60 echogram from the ultra-short baseline, otherwise known as our USBL, and that is how the ship and ROV monitor their distance from each other and monitor their distance from the sea floor. So we are aware of exactly what that is, and that's how we know what that noise is filling in there.
Thanks, Mike.
So ironically, in this case, it really is noise and not just noise.
That is right. Yeah. Ironically, would you know, in sonar, what is noise to some is signals to other.
Right.
So these deep scattering layers were initially discovered because people were trying to use sonar to map the seafloors and other Kawhiti. And all these layers are getting in the way.
So what depth are we going to right now?
We are going to 900 meters.
900. So 900, and then it's the 7.
And then we'll move to 710, 600, 450.
Got it.
And we'll keep an eye on the EK60. We can only do
See
so
if it
much to chase the
changes.
layers around.
Yeah, right.
But we'll see what they do.
So Amanda, we saw this morning on the ROV's transit down to the bottom. We saw avoidance, at least in the lower layer.
Yes.
And does that have a specific name?
We just call that avoidance.
Avoidance. Okay. What about the layer itself? You said it was comprised of different creatures in the upper layers.
Yeah, so generally the upper layers are composed of some of the smaller zooplankton and fish. These are a lot of the animals that make the diel vertical migrations, the daily migrations up to the surface at night. And then the larger fish, the dragonfish, chiliolids, what are they called? Viperfish. A lot of these larger ones, these are the ones with the really big mouths that we think of as classic predators. They tend to hang around a little bit below that upper layer, and they can kind of dart in and out and feed on those animals that are making up the core layer. A lot of those animals that live in those lower layers and the more predatory fish down there are likely highly undersampled. So we think when we compare what we can collect with nets, doing a traditional net trawl, and what we see in acoustics, we think we're only collecting about 10% of the fauna that are out there. And what we're missing primarily are those larger predatory fish that can swim a little bit faster than the other animals. So you can see why it's tricky doing work down here, because there are things that can avoid the nets, they avoid the ROVs. Acoustics, only tells you so much information, but at least you get to see where the animals are.
Just got to drain the ocean through a great big sieve, and
exactly.
then we'll get it all.
Exactly. Something back there.
Yep.
It's either close Kawhiti or distant Siphonophores. There's a little jellyfish, little dark ones. Let's see if So our colleague, Dougal Lindsay, had to hop off for a little bit to recharge his Wi-Fi device. This is one of the, the wonders of telepresence, is the many different places that people are tuning in from. I've definitely done a lot of these dives in the middle of the night from hotel rooms, but I think my strangest telepresence experience was being on another telepresence ship and calling in and leading some Midwater transects from there while attempting to stream the video on my cell phone because we were close enough to shore. They didn't want me streaming video on the ship because they needed the bandwidth to get their video off the ship. So we're always juggling some things to make this happen, but it's a lot of fun to have so many different people tune in from so many different parts of the world. There's a Siphonophore, a little long one off in the distance.
That must be some good phone that Dougal has.
I know.
This will be good. We'll just be at the bottom of this blue layer.
Yep. Interesting. Can we zoom in on that again? Awesome, thank you. All right, so we're
Oh, there goes
getting
a
really
shrimp,
close
I
to
think,
900
just went
meters.
by.
Nice.
There's something else.
Yeah.
A little copepod. It's a good sign.
So it looks. So it looks like excellent. So we are
Copepod or
starting our
something right in the
900
center.
meter transect.
There's a jellyfish top, just went off the top middle of the screen.
Nice. See if Dougal had an idea.
Hope so.
Yeah. we'll see what Dougal says.
Yep.
Thank you.
So that had a really beautiful dark red color.
Yes. Do you want to tell us a little bit more about why things at this depth are red, Astrid?
Sure. So In the ocean, red as a wavelength comes out the quickest, so you get basically no red light at depth at all. And so being red in the deeper ocean can be a form of camouflage because organisms actually aren't adapted to see red light, and so they won't be able to see red organisms.
Thank you so much. All right, is there something here towards the top middle?
Polychaete.
Awesome. This is a Tomopteryd polychaete worm. These are one of my favorite animals to see in the midwater. They're so bizarre looking. So we think saw some polychaetes today on the seafloor.
Yes, we did.
Yeah, so polychaetes, they all have these segmented bodies, but they can take a enormous number of different morphologies and shapes and sizes. So these are one of the more common pelagic ones. We do see some others, but really neat animal. So if anyone is looking at the EK60 data, I don't think that's just that.
Yeah.
But
Oh,
there on
there
the
we go.
upper left, yeah.
Siphonophore, maybe?
Could be.
It's got to be, because I've typed it in.
That is not a siphonophore.
No, oh well. I can erase
I
it.
can tell you that much, and I otherwise have no idea. See, this looks to
this
me
could
like
be a
what you
pseudomorph. Yeah. It's a mixture of squid ink and mucus.
Yeah.
Used as a getaway device.
Yeah. So unfortunately, I thought we were going to have Heather Judkins online, who's a squid expert. Hopefully, she'll tune in for some of the later midwater dives. But she said, we kept seeing things and just going, oh, it's goo, oh, it's goo. And she was like, no, it's that's how we know the squid were there. Yeah, it looks like a Keatignath down there. It'd be great to get a nice zoom on that. Oh, that's a fish. Nice. Yeah, and this is a cyclothone. Awesome, thank you. So if anyone is looking at the EK60, we're right at the base of the lower layer now. I'd say we're definitely starting to see more animals. There's another of those little jellyfish. Do a quick. Don't see it anymore.
Oh, yeah. There we go.
Nice. Huh.
You got this. You got the something, right?
Yeah, exactly.
Wonder if it's a molt of some crustacean or something.
It's possible.
Yeah, I don't know.
Really hard to say.
Yeah.
But I think that was, we'll pass that along to Dougal. Dougal's going to get a whole pile of things to identify after this. My background is in midwater fish off the coast of California, so I'm slowly learning the gelatinous fauna and really enjoying that, but definitely not as expert as some of my colleagues.
Is this something right in the center now, or just to the left of center? Yep, Kawhiti again.
And if it's possible to get any closer, yeah, they have a pretty good escape response, so it can be challenging to get a really good image.
Yeah.
There,
there we go. She warned you.
Got some really good footage of a Kawhiti being a Kawhiti. It's a Kawhiti or an arrowworm is the common name for it, and they have these jaws around their mouths that open up really wide to the side, so they can eat things that are really large compared to their body size. So they eat pretty much everything that's out here that they can get their mouth around. Well, thank you. So I talked a little bit earlier about how the cyclothony or bristle mouths, the species that live in shallower water, tend to be small and more transparent. As you get deeper and deeper, they get darker and bigger. And it's because they don't need to hide so much. Kawhiti have a very similar kind of pattern. The ones that you collect up near the surface tend to be really rather tiny and again, transparent. As you get deeper, you can see much more pigmented species of them and quite large species. See similar patterns with copepods and all sorts of other taxa. It all has to do with being able to avoid getting eaten.
Something right in the center? I don't think so. Looks like a fish hook. I think it's just a mucus string.
Yeah, so this is likely leftover from a siphonophore, it looks like to me, but most of the
Yeah, I think that's a good guess.
And we'll likely start seeing more siphonophores as we get into those shallower later layers. Excuse me.
Another copepods.
Yeah. Oh, what is that? This looks different.
Yep. Is that a little squid?
Fish?
That might be a little squid. Or maybe it's, yeah, I don't know. Fish, squid.
It looks fish to me, because I'm only seeing the light on one edge of it.
It's like head up or something?
It looks head up and belly towards us.
Oh, I see.
See what I'm saying?
Yeah,
so this,
I do, yeah.
Many of the fish that live, this is definitely fish. Yeah, if we can keep trying to image this, that would be fantastic. So most of the fish that live around these depths, what is it doing? I don't even see photophores. I think it's just reflecting.
Siphonophores would probably be underneath, right? We're looking at the side.
Yeah, but they're often kind of along the belly and along the side too. Do not know what kind of fish this is. Astrid, do you have any ideas what kind of fish this is?
No, I'm kind of stumped here.
I know. They're getting some incredible imagery, which is great. That's awesome.
I think that side shot will be really helpful. We got it kind of from the side before. I think that'll be useful to ID it later on.
Yeah.
It's got a pretty wide mouth. It's almost like a, I don't know, larva.
I lost the feed here at home for some reason, so I can't see, but I'm listening to what you're saying.
It's almost like it's a tiny, like dragonfish, but up in the water column. Well, I know it's not, but because it's got that really wide mouth, it opens really far back. So you know, like the Benthosaurus, that's what I mean, that sort of thing, but the really wide mouth.
Yeah.
Well, yeah, that was probably one for Bruce Mundy too, because he knows a lot of the midwater fish as well as the benthic ones.
It's really holding itself steady.
It is, which is very unusual for the fish. They tend to try and get away from us.
He's starting to go into the horizontal, though.
If we can get any good side shots, that would be great. I know, it's hard. Just ask it to keep spinning around a little bit. Great, thank you so much. Yeah. That's awesome. That was really cool. Okay. So we're going to go to 710. Yes, please. Thank you.
This will be exciting. Here we go into the layer.
I know. Hopefully the layer doesn't leave us. Oh, that's a nice jellyfish. Oh, what is that?
Another Pelagiatheria, I think.
Was it? Cool. Do you know anything about their distributions, the Pelagiatheria?
No, and I've never seen them until, you know, these cruises.
Yeah.
It's amazing.
I mean, I don't think I've ever collected one. So I don't know if it's more of an oligotrophic or tropical kind of thing, because most of my work is eastern boundary. They're really neat.
Yep. I'm impatient.
Yeah. What is that? Oh, it looked like Siphonophore bits. We're definitely getting into a
There's a
lot of
Medusa.
stuff here.
Yep.
Yeah, there's a.
That's a little. That's a Hydra
That's a
Medusa
Hydra
for
Medusa,
sure.
yeah, yeah. There's something shining off there on the central top. Fish, I think, on the bottom right. So you can tell we're definitely starting to punch through this layer, starting to see a lot more animals. There's a Siphonophore. It's moving down the screen. Oh, yes, sorry. So now it's 710, 600, and 450. Yeah, so we changed the last three. Awesome, thank you. Yeah, sorry, I forgot about
I don't mind helping you.
that.
Yes, your name is Watch Lead for the foreseeable future.
I finally got used to being so very loud. Another Ketignath likely going through the screen. Something going by.
There goes another one of those red jellies.
Nice. Let's try another Ketignath. There's definitely a lot more animals here as we get into this layer, which is great. Survey 1, can you hear me? Can you hear me? Can you take another screenshot, please? All right, Siphonophore off in the distance there. of that.
I want to see that one.
That is. That a Steve Haddock thing? I don't remember. Someone had that in their presentation or something.
Oh, sure.
Might be Google, actually.
Here's that radial Aaron thing again.
That's a strange one.
Yep.
That we did not see.
I think that's what I was showing you, the image of just without its gelatinous stuff underneath it. Yeah, it had all these little structures.
Amanda, can you hear me?
Yes, I can hear you. Are you back with us?
Nope, I put my phone on speaker. So I was things weren't working right, so I'm actually on the YouTube channel.
Okay.
The dive, so I've got quite a delay, but I was able to look back at that fish. That
Yeah.
Got some good images of.
Yeah. Do you have any ideas?
This probably isn't right, but one suggestion might be
Hold
that
on,
it's a type of whale fish. One of the red ones, and that'd be the family Barbaricity.
Okay, starting transect.
The rounder lobes on the caudal fin.
Starting the next transect, Chris.
What's that?
We're starting the next transect.
We're starting our next transect. We're at 710 meters.
No, I know that I was just looking back at that fish, seeing if I could figure out what it was.
Yeah, no, that's great.
Suggestion that it looks a bit like a whale fish Barbaricity, but I could be way off.
Here's something lower right, left.
Yeah.
That's my left, yeah.
That is another larvation.
Okay.
Got a quick look on it. Let's just look at it for a minute here, yeah. You can really see those
kind of a classic
chambers,
one.
yep.
Yep.
I don't see the larvation in there.
Yeah,
so
not
I think it must have
that's
just,
what that is.
no, I think it must have recently abandoned its house. They collapsed pretty quickly, but you will see recently abandoned houses. Thank you for that. Yeah.
Space hasn't opened up, so I guess that's good. Oh, here we go.
There we go. So
That's
this is a
ctenophore?
ctenophore. Again, we saw a ctenophore earlier, and it was really transparent. But you can see these same 8 rows of combs or teens that are along it that are common to all ctenophores or comb jellies. This one has these 2, so the ctenopods have 2 really long trailing tentacles, so you can see hanging off it. And you might be able to see, I can make it out faintly, that each of those tentacles kind of branches a lot. So this allows it to sense a huge volume of water, even though the actual organism is pretty small. Thank you so much.
I like to describe it like long line fishing.
Yes.
A long line and have all the hooks coming off of that line. There's a couple of things that just went.
Just on the right there.
Oh, and here's a Medusa on the left, just went off the left of the screen.
There's so much here.
Yeah. Sorry. I thought we'd go for the Medusa.
It's up. Yeah, there's a fish swimming through straight ahead.
Eel from the top.
Yeah. Be great if we could get any closer.
It's like a
Is this a snipe
duck-billed eel or something?
This looks snipe eel, I believe. So this is great. Astrid, do you know much about these guys? Is she still on? Are you still on, Astrid?
She's in the chat room. She says snipe eel.
I'm here. Nothing in specific. I just know from the trawl samples that we always thought it was really funny that their mouths are they're split like that, and we always thought it looked like they would ram into a wall or something. But that's actually their morphology, their mouth morphology, which was interesting.
Yeah, they are very strange-looking animals. So that kind of eel-shaped body form is really common at these depths, and similar to the ctenophore we just saw that had that, almost net of tentacles hanging behind it. For a fish to be really long and skinny allows it to have a really long lateral line and sense a lot of the water around it. So there's not much light here for them to see, so they have to use other senses. What?
Siphonophore?
Yes. It looks upside down and weird.
Yeah, it's weird, huh?
I don't know what it's going
Oh, no, wait. This is Selp, Colonial Selp, isn't it?
Oh, and that... no, it's...
Oh, no, yeah, it's a
Siphonophore. Yeah, it was
Siphonophore.
just
Wow, I don't recognize that one.
You never see them in that orientation. That's cool. It's
yeah,
a really...
that's a good one.
Beautiful barrel. So Siphonophores are colonial relatives of jellyfish. So just like the corals that we saw earlier today, our colony is made up of many different polyps.
Are you sure that's not a Selp? That sure looks like a Selp to me. And these are, they produce chormidia. So these are long strings of reproductive structures.
I've never actually seen that.
Because these are the muscle rings.
Yeah, it's doliolid.
Yeah,
the rings go all the way around, so it's a doliolid, not a Selp.
Well, sorry, I put them together in the Eurochordates. That's what I think.
But it looks like swimming bells. I'm really confused.
I'm gonna.
We don't have.
I'm gonna look it up for you.
Yeah, please do. That absolutely looks like a doliolid at the top, but the rest of it looks like swimming bells to me.
Yeah, no, I'm sure those are reproductive structures that they trail their eggs out behind them.
Yeah, and they can be paired like that because there were sort of two
I've never
rows
seen them
of
paired,
it. Yeah, that was cool.
There's a lot of things I haven't seen in the water column.
Yeah.
Here, like this.
That was really cool.
Yeah, yep, totally. So Scott just pulled up a picture of it, and they make those pairs. So we were totally off.
We got there.
We got there, but a completely different phylum there. So these are tunicates, and I think I've actually never. I've collected doliolids, but I don't think I've ever actually seen a doliolid from an ROV, and that was really quite incredible. Something in the middle there. There's also, yeah, we can go in for that blue thing. The other one was a Ketognath. That's a hatchet fish. Yay! So these are called hatchet fish. They're a very common midwater fish. Number of different species throughout the world. some folks have done some really interesting modeling on these fish. So they're really compressed side to side. So if you looked on it head on, it would just kind of look really thin and skinny. And if you shine different light on it from different angles, the way it would be in its natural environment, so not us here with an ROV shining a bright light head on at it, they camouflage from every single direction. And these guys do have photophores along their stomachs, and also the kind of dark back if you were looking straight down on them. So really good camouflagers. Not very fast swimmers, which is great for imaging them. Thank you very much. That was great. We're seeing everything.
Yeah, there's something dark right in the center of the screen. It's going off to the bottom
looks
right.
Probably fish to me.
Okay. No worries.
Go.
No worries.
No worries.
There's
There'll be more.
Else.
Yeah.
Yep.
We're
I'm glad you know what direction you're going.
If we were driving the ROV, it would be all tangled up. There's something in the upper right.
Oh, is that a jellyfish?
Yeah.
Siphonophores? For sure.
Yeah. So these guys, look like one of the kinds of jellyfish that will drop their tentacles very easily. And that's basically a distraction for predators. So if something tries to come after it, it can drop its tentacles, which are quite a bit larger than the actual kind of body of the animal as well.
So was it a siphonophore or a hydrozoan?
I think it's a hydro medusa.
All right.
Because that had that band around it. Looks Keatignath again. So we don't need to chase it too much. Thank you. Yeah, we're seeing so much here. You can see how diverse the community is at these depths.
Dougal's asking if the snipe eels had 2 black bands on the tail. I sure don't remember.
I didn't notice 2 black bands on the tail of that snipe eel, but it's possible.
I didn't notice any black bands either, but I think I'm going to try to go back in the video and see if I can find out.
All right.
There's something right in the center. I don't know if you can zoom on it well. Yeah.
Okay, so we are at the end of our 710 meter transect. This is probably the snipe eel again. So
oh yeah, look at that.
Could be the
It's
same
got
one
the
or a
black
different
bands.
one. Does it have black bands at the tail? I don't see black bands. Oh, there's sort of one.
Thanks, guys. That's
yeah,
good.
thank you so
That's
much.
good.
Thank you so much. That was our little bonus observation. For those on the phone, we're headed up to 600 meters now.
Hi guys, so I just looked back on YouTube at that snipe eel, and you can't really tell whether it has black bands on the tail or not.
Yeah, it looked like it could have had kind of some thick fuzzy ones. So there's a.
That could be some misses.
Yeah, is it possible to zoom in on this jelly? No. Okay. No, that was not. That's okay.
That's a
That's
pteropod
like
or a
a
heteropod.
gymnosome, yeah. Cool, thank you.
I'll tell you, there's an awful lot of things that needed to be added to the.
I know. We'll go
column.
back through and do annotations properly for these. The one upside of doing shorter midwater transects and fewer of them is that you can annotate really thoroughly afterwards. It's not the same time commitment. A lot of our collaborators are on the east and west coast, and it's pretty late in both those places now, so to follow up with them. I feel like we need to close our eyes while we...
Yeah, so we stop saying, yeah.
Yeah, I know. I can't do it, but.
I think there was some misunderstanding there about who is supposed to close their eyes. Yeah.
Yeah.
So the South Doliola thing, they're all thalassiaceans to me, and so
yeah. Yeah, so
the
Souths, the bands don't go all the way around.
Oh yeah, no, I know the difference, but I just can't
adorable.
get the name at the time, you know, when I need to.
It looked so Siphonophore to me, the rest of it, that I couldn't, I was like, I see the bands, and I just can't get
So the interesting thing is you have to believe yourself, right? You said the gas bladder shouldn't be down below. They should be up on top, or the gas-filled zooids.
Yeah, and that was Dougal chimed in on the chat room and was saying there's a lot of mimicry, out in the pelagic zone, and so it's really common that we mistake things for each other. And there's on the last one of these dives we did, and I would probably have a hard time recognizing this as such because it's such a good mimicry. There's a Siphonophore that looks like a Medusa.
Really?
So it's just kind of taken on this form. And it's something that, we haven't even known about for very long. So we don't have the tools to get at it.
Well, that was a beautiful doliolid, I must say.
That was gorgeous. That's the money shot. That was gorgeous. Is that a Siphonophore probably?
I looked too late.
Yeah, I just got that.
There's another Siphonophore.
So I'm hearing some chatter from our pilots about the different zones in the ocean. So the kind of classical zonation is 0 to 200 is epipelagic, and so those are still kind of in the influence of sunlight and photosynthesis. So most of the primary productivity occurs in the epipelagic zone, and everything living below that is getting its food somehow from there, or on the seafloor in rare cases from chemosynthetic environments like vents and methane seeps. From 200 to 1,000 meters is mesopelagic. A lot of our transects have been in the mesopelagic zone, but our first two were in the bathypelagic zone, which is 1,000 to 4,000 meters. So we did a transect at 1,500 and at 1,200, so in the upper bathypelagic. And then below that, we get into the hadal-pelagic. And that's where you start getting into the pelagic zone up above trenches, and that's where we suspect some of the most undiscovered biodiversity on the planet is probably in the bathy and hadal pelagic zones because they're so hard to get to. The hadal pelagic in particular, you don't want to be dragging a net that's, miles long in a deep, sharp-edged canyon. So really tricky stuff to study. So it looks like we're doing a quick pilot change before we get started with our 600-meter transect.
I see Dougal's writing in the chat room that this dark pigment that you see in a lot of these midwater animals that turns them red, the porphyrin pigment, he says it turns poisonous when it's exposed to sunlight, so they die if they get too shallow.
Oh, that's very interesting. I did not know that.
It's crazy.
Yeah, that is.
There's something drifting through.
Could be a fish.
Oh,
We're not
we
starting.
weren't
We thought
watching.
we were starting now. Okay. Thank you.
There's something red just went off the top of the screen.
All right, so for everyone on shore, we are just starting our 600 meter transect.
Something in the
This is
center,
our second
right
to
of
last
center.
transect. This is looking Kawhiti, but it could be a fish.
I'm going fish.
Yeah.
It's another snipe eel, like upside down.
Yeah, that's what.
Yeah. I don't know.
Coloring's a little funny,
Yeah,
but
it
I
actually
think
just looks like
that's
a mucus string with a
but
wiggly
yeah,
tail.
it's clearly swimming, but it does.
I just see there's some real narrow point at that end.
Yeah. All right. Thank you.
I'll just call that unknown.
Yeah. We end up with a lot of unknown unknowns in our midwater annotations. Okay. That would be great. Yeah.
Here we go.
Nice.
That's
Now
a
we have a
siphonophore.
siphonophore. This is
Yep.
a real siphonophore. So go back to my siphonophore spiel that I started earlier. These are colonial jellyfish, which means they're made up of different individual zooids. And they have different types of zooids. So they have ones that are used for swimming, ones that are for eating, ones for reproduction. In theory, they can all survive on their own, but they don't survive for too long without the other functions that are provided by their cohort. Screenshot. So I'm looking at the acoustic data on the EK60, and you can see that the scattering layers are starting to make their migration. They're starting to come up for the night. Typically the smallest animals come up first because they don't need quite as much darkness to hide up there, and then the larger animals will follow.
There's a little Medusa just to the right of center. All I can say, it's Hydra Medusa.
Yeah. Yep, we're seeing that too. So I'm thinking this is going to be another Keatignath. I'm going to play the Chris Kelly game and try and identify things before I get to them.
It's a good game.
That is.
Sure looks like it to me.
Yep, that's definitely Keatignath. Nice name, thank you.
Nope.
So we just do this, and then you can highlight that. All right, so we've got a Siphonophore in view here. This is another one of these colonial jellyfish. It's a really long, rope-like species here. Siphonophores can get extremely massive from end to end, dozens of feet long. Okay.
It's all those heavy rocks in the starboard rock box. Dougal got excited by that last one. Wow, Paralepid.
Something. Let's see. Okay.
Is this just another Ctenophore, or is this something else?
Can't tell. Can we zoom in on this in the lower left? There's a Keatignath at the top. There's a fish coming in. This is Siphonophore. If we could take a quick look up and see if that fish is still there, yeah. I would rather try and get the fish. Oh, yeah? Oh, yeah.
Squid.
Squid, nice.
So you thought that was a siphonophore?
Yeah, I
Okay.
do. And then some kind of fish. I couldn't get a good look at it.
Yep.
That came through. Yeah, off on the right there.
So same
thing
thing.
we saw before.
Yeah.
I don't know what is going on there. All right. Do you think there's a fish? Weird. All right, Ashton thinks that was a fish. All right, so yeah, 450 meters for the last one. Let's see if that still makes. Yeah, it should be good.
I can't leave now.
No, you
Finally
can't leave
getting up
us.
into the good stuff.
Of course, it keeps moving away from us because of the migration. Siphonophore. It's a suggested shrimp. They're a little shrimp of some sort, copepod. That's good. Good timing. Nope, we're just getting to 450. So all right. Are we starting the 450? All right. So for everyone home, we're just starting the 450 meter transect. So what's been interesting from watching the acoustic backscatter is that
Here's something coming on the
the
left.
layers we saw, that's interesting. The layers we saw when we first did our plantings, this is another siphonophore. These are Physonecked siphonophores, so there are a few different types of siphonophores. These ones have a little gas bladder at the top. There's still a great amount of diversity in the sizes and shapes, even just within the Physonecks.
You can see all the little bells were pulsing.
Yep. So the Physoneck siphonophores are actually quite indistinguishable from fish. Of course, not visually. They look really different than fish if you just look at them. But if you're looking at acoustics, because those siphonophores have a little gas bladder at the top of them, and a lot of fish have a little air bladder in them, they both have a really strong acoustic signal because air is really different density than water. So it can be challenging sometimes to sort out whether your signal is coming from siphonophores or fish, which are very different ecologically speaking. Being able to do things like this, actually go down there and look at what we're seeing in these layers is one really useful way of starting to sort that out. And one of the reasons why we can't just use acoustics alone to survey for organisms down here. Gives us great spatial coverage, but terrible taxonomic resolution.
Is this something at the top center?
There's a little
Yep,
copepad,
there's two things.
but let's go to the top center one. Yeah, let's zoom in on that. It's probably. Yeah, there we go. So this is definitely a Bristle Mouth, and we saw some of these earlier, and I suspect this is a different species because it looks to me to be lighter in color. So you can see along the belly it has little photophores. It has light-producing organs. Again, very common feature of organisms in the midwater. Great. Nice.
Real transparent body.
Yeah. So these guys, when you collect them in a net, they have really large mouths, like many of these other fish that we've been seeing, but their jaw bones are so thin and fragile, and their gill rakers are also really thin and fragile that typically when you collect them in a net, the whole head portion gets kind of mangled. They get tangled up in other things really easily, so it's really unusual to see an intact cyclothonia like this.
There's a lot of mucus in the ocean.
Yes, there is a lot of mucus in the
here's
ocean, and even this is probably another, we think maybe radiolarian, or some other type of single cellular organism.
Oh, wow. Yeah, this is not what I was showing you before.
No. Is there something below it?
Yeah.
Why not? Oh, yeah. Yeah. Masters of camouflage.
I wonder if that looks like the top of one of those kind of pyramidal siphonophores.
The Calicopheran siphonophores?
I don't remember what they're called. I have a figure in my head.
Yeah.
Which is no help.
People always call it rocket shaped.
Yeah.
The top of the Calicopheran. Yeah.
So that's what that looked like.
It's possible.
There we go.
There's a suggested shrimp. These are quite common. They have these antennae that kind of fold back on themselves. But again, we're seeing this similar kind of structure that we've seen all across the many different taxa that we've been seeing today of having some sort of elongate appendage or elongate body form. So again, this allows them to be able to sense a much larger parcel of water than they otherwise would be able to in this dark ocean. Great image. They're really cool.
So you said suggested. Do you know Lucifer, the genus Lucifer? Is that a suggested? Do you know that?
I don't
Just
know.
I know that the crazy eyes, the larval stage.
Know.
He's paddling and paddling and paddling, not getting anywhere. Save his life.
So it might be feeding. It might be creating feeding currents with its little legs.
What's that? There's a little.
Oh yeah, right above it.
Yeah, just went off the top of the screen.
Yeah, if we look up and to the right, perhaps there. So now it's floating down. I don't think we're going to be able to get it.
It's really small.
Yeah.
I'll bet it was a Medusa, you know, the 4 gonadal pockets.
Yeah.
Oh, there it is. He's back. It's right there on the right side of the screen.
Yeah, just about to drift off the right side of the screen. I don't know. Keep leaning in closer, like it's really
it's
getting
gonna help.
much
Yeah.
closer.
All right.
Yeah,
Not
so
a Medusa's
no.
home. Huh. Is that a larva? No. This one is like some echinoderm larva or something.
Oh, I think so.
If one ever wants to really truly show their ignorance, they should come and sit and do this. There's a lot we don't know about what's in the water column.
Yep. So we just got a message from Astrid that she's going to have to be
There's a siphonophore in the
taking
lower
off
center.
in a few minutes. Yep. So Astrid, I just really want to thank you for joining us and you know, helping explain so much about the fish and other animals that we've been seeing. It's been really great to have you join us, and I hope you're able to continue to do so for the other midwater dives.
This is a copepod, maybe?
Yeah, thank you guys for doing this. This is really neat. I'm definitely going to be tuning in for the rest of the midwater column stuff too. So
excellent.
I'll be around.
Excellent.
Fish.
Yep. So this is another
Nice.
cyclothone.
Cyclothone.
So yeah, Astrid, you were suggesting that this is Cyclothone pallida, and probably a different species from what we saw deeper. There are slightly different species out here than there are off the west coast. So I'm still gonna need to look back and sort out what they are. For some of the Cyclothone, you have to do things like look at and count gill rakers to tell between different species, so it's almost impossible. But so they're good questions for Bruce Mundy and some other colleagues I know who are expert in these fishes. This looks like schmutz.
But it's a really good view of it.
We need
it's
a category
really good
for marine schmutz.
Yeah, a discarded house or something.
Yeah, it's like collapsed larvation house. But
you got it really well.
It's a gorgeous image of it,
Yep.
and it's very important. I do see these two little red spots on it that look like they're probably something, either copepods or amphipods or something that are actually feeding off it. So that's actually quite cool to see.
All right. We're going to make an observation of that because we are hard to get that. Yep. There's Kawhiti now.
Yeah. I'm thinking Kawhiti again. But it could be a fish.
Yeah. Looks. I don't really see Kawhiti tip. Yeah, it's a fish.
Yep. Nice. This looks like the same kind of fish we saw earlier.
That whale fish thing that Chris thought? Is that what you mean?
Yeah. Can we zoom in on that guy? There's so many fish here.
That's a cool one.
Yeah, so this is, I believe this is a barracuda.
Oh, good. I was going to say, it looks like a barracuda.
That looks really similar to the one we saw before, don't you think?
You think it's the same one?
I don't know why
it looks longer to me.
Yeah. The body structure and things look similar, and the tail is still that kind of rounded lobate tail. I don't know.
Yeah, no, it's definitely possible, and it's just getting at it from a different angle.
Come on, one more cool thing.
All right. So for those on shore, we have 1 more minute left of this transect, and then we're going to be making our way back up to the ship. I'm
Where our
planning
work
on
will continue.
staying in here and seeing what flies by on our way up.
It's another little
Like
Keating
lower
mouth,
left,
maybe.
yeah.
Not worth chasing. This looks like it has tentacles right in the center.
Yeah, this looks. See, they're going to be probably a Tinophore or a Narco. I think it's a Narco Medusa. I think there are 4.
Oh yeah.
Yeah, if we can zoom in.
Oh, nice.
Ah,
Yeah, I think it's a little narco. It's a weird one.
yeah.
Really small bell.
Yeah.
What else would it be?
Possible it's one of these strange single-celled things with appendages that I can't remember the names of. I think is more likely.
Okay. I'll put unknown.
Something. An animal.
Undetermined.
Metazoan. That's a fancy way of saying unknown.
Might not even be a metazoan if you're saying it's single-celled.
That's true. True.
All right.
Are they asking us?
No. I don't think so.
Okay. So I'm like, yes. Another Siphonophore.
What's this down here? There, that's a Tinophore.
Yeah. There's a sedipid Tinophore. Those long trailing tentacles. You can see those teens rapidly firing away. These are the little tiny cilia. And these guys don't actually bioluminesce like many of the other animals down here. A lot of people see that light refraction coming off them and think that's what's going on, but this is really just the light reflecting off of those moving teens.
Swing mouth forward. He can hear you. You can see what we were talking about here before with equating it to long lines. You can see the long tentacles, each with small branches coming off. And each of those little branches is basically has these sticky cells called coloblasts that it can capture prey with.
All right, so we have a bonus transect today, which I'm totally stoked about. And thank you very much to our ROV team for offering that and not taking the early break. So we're going to head up to 300 meters. As we've been doing these dives, the sun has started to set and the deep scattering layer has been making its migration up towards the surface. So this will allow us to hopefully intersect with some of that layer as it's coming up, which is awesome.
Yeah, good stuff.
Sure. So as we make our way up, it's a good time to thank the ROV team. I know that the Midwater work is really challenging. It's a very different way of operating and requires some pretty precise controlling to be able to actually, stop, get the nice zooms on the animals and follow things around. So this kind of work really depends on a pretty large team of people, including the scientists, our scientists back on shore, the whole ROV team, our ship crew, everyone involved. So it's we're greatly appreciative for all the support that we get to do this work. I think that's a Medusa. That could be a Solomyces. That, yeah. I'm gonna just put a note in here, even though it was quick. What do we have here? Oh, really? Is that how you spell it? Solomyces is just one S? Okay, so I had it right the first time. Okay, 3 S's. Oh, thank you. That could be another one. That's the time when I'm supposed to close my eyes. There's nothing to see here.
Well, at least I think we're going to be in this blue at least this time.
Yeah, hopefully it doesn't move away.
Right, yeah, well, trying to get a bit ahead of it.
Its way up, but it's a fun time of day to be doing work because everything's really active. All these things that are migrating up to the surface are really hungry. That's why they're going up there, is to get to the food that's dependent on all the primary productivity that's up at the surface. So, everyone's, it's like us at mealtime. We're all buzzing around and elbowing each other. It's kind of the same thing with these critters. Another Ctenophore. Oh, not Ctenophore, Ketognath. Yeah, sorry.
So start with a C. Maybe another one.
Try to get some annotations in. That's what it is.
No, that's too long, huh?
Mm-hmm.
There's another one of those little, looks like a die,
Yeah, it did. On my other computer, I have a fish guide that I think I have. I think I might be able to figure out what that was. Okay, bye again. Fish. Oh,
Yep.
fish.
Swimming towards us. Attack!
Hey, buddy.
Coming in hot, as Mike would say.
That's getting a. That's a fish.
Yeah.
We've seen a surprising number of fish.
Yeah.
I'm quite pleased. All right. So for anyone back on shore, we're getting close to our 300-meter starting point for the final bonus round of our Midwater transects. It's weird.
It's a glitter party for the last bonus round.
I know. This is more fun than I thought it was going to be. I have to decide what's more awesome, deploying Argos through Midwater transects. Okay. Excellent. So we are starting our 300-meter transect.
Nope.
And it looks like we're still pretty nicely in a layer there.
Maybe this stuff is what's causing the layer.
Yeah. I know. I'll leave it. Oh my God. Thank you.
Nope. Just mucus.
Almost there?
Yeah, that thing that came zipping in. And
Yeah.
yeah.
Looks like
Here,
here on the left.
yeah.
Something shiny.
Must be a
Definitely
fish,
a
huh?
fish.
Yeah.
Definitely a fish. Beyond that. Huh.
Interesting circles he's
I know.
swimming in.
It's like chasing its tail.
Yeah.
Really.
Didn't you tell
Looks
me
like
the other
he's
day that
changing
things
direction.
did that to fool, make themselves look bigger or something?
Yeah, this is okay. Thank you. That was, I don't know what that was. Yeah, so that wasn't exactly the curling behavior, but so this was probably more of a sensory kind of thing, just trying to, snake its way through as much water as possible. When you see the curling behavior of the fishes, they kind
It's
of
up
curl
at
up
top.
like a cat.
Oh, he went off.
That's
No,
part of
that's
that's a
a-
Siphonophore.
yeah, that's a Siphonophore.
Can see the combs. It's not entirely intact, but So Scott was asking you about, we were talking about curling behavior. There are a lot of animals that curl up in the water column. They tend to just kind of hover there, and they're basically trying to camouflage as a jellyfish. It makes them look like a medusa. So they'll curl up and just hover in the water column. There's a fun paper about that.
Something in the center,
Yeah,
right
zoom
center.
in on that. This is Siphonophore, I think. Somewhere, yep, there's the swimming bells are at the lower left, so we can kind of run along this down towards the lower left. This looks like it could be a Chalicophoran siphonophore. So this is a different type than I think we've seen so far. So these do not have the floating bell at the top of them.
Pneumophore?
Pneumatophore is
Pneumatophore.
yeah. So if you're looking for fish in echo sounders, the Chalicophoran siphonophores are not so much of a problem as the...
Got it. They don't have fake swim bladders,
they
is
don't
that
have
it?
fake swim bladders, exactly. That freaking out over there, right in the middle.
Yeah, I
There's
can't tell if that's
zooming
another
around.
fish or a keto.
I think it's keto. Yeah, I think that was another ketognath or arrow worm. So I have some colleagues off the West Coast who were using passive acoustics to listen for whales and dolphins off the coast there. And every evening and every morning they were hearing this for, half hour or so. And they believed that it was the sound of all of these organisms making their dial migrations at the end of the day and in the morning. So it's kind of one of these true exploration stories, because they went out there to listen for whales, and then they've ended up doing a lot of follow-up work to try and understand what the deep scattering layer is there and whether that is synchronous with the sounds that they're hearing. That's really interesting.
Well, there's something right in the upper center.
It's to the left now.
Yeah, just went off screen to the left, about the middle of the screen. And now it's in the center now.
Yep, and now it's getting right. Nope. It went too far to the right. Yeah.
Yeah, it's okay. It's gone. It went off the right.
It's hard when most of your targets are see-through.
Yeah.
You guys have been doing an awesome job. Yeah, see what that is.
Ctenophore? I was reflecting that way.
Yep, definitely ctenophore. This looks like it's lobate.
Might just have the tentacles retracted, I think.
Yeah. Yeah, yeah. So it's another sediped ctenophore, but the tentacles are kind of tucked up into the body instead of trailing behind it, like some of the other ones that we have seen today. Oh,
There's
something on the lower right, moving to the center. This is a siphonophore.
That was your last bonus.
All right. Great.
Thanks, ROV team.
Thank you so much.
Yeah, thank you guys. This has been awesome.
Thanks, Astrid. I'm really glad you could join us. It was great to have you on the line. And have a good evening at home.
Thank you. Good night, guys.
Good night. All right, so for anyone back at shore, we just finished up our final bonus transect at 300 meters, and we're now going to be making our way up to the surface to recover the ROV. That was really an incredible set of transects that we had today. We saw, multiple different kinds of siphonophores, several different kinds of jellyfish. We saw a doliolid that was reproductive and had small chains of baby doliolids behind it, which is really incredible, something I've never seen before. Many fish, including several different kinds of fish, which are often quite elusive when we're doing ROV transects. We saw copepods, we saw shrimp, and I'm sure I'm missing more things. It's really been an awesome set of transects, first observations like this in this part of the world. So yeah, anyone who's still tuning in, really glad that you could join us for this.